Chapter 15, the special senses. So we're going to look at the special senses, which and general senses. Special senses, like you see in the figure at the bottom, in this first figure, these refer to neurons that detect light, chemicals, sound, that are present in our environment, and then convert or transduce those stimuli into an electrical signal. As you can see in this figure, taste, light, sound, head movement, and smell. These are your 5 special senses. 4 of them, taste, light, sound, and head movement, those are detected by special receptor cells. Okay. Smell, another special sense, or olfactory, those are received by receptive endings of actual olfactory neurons. Okay. Now your general senses at the top of this figure, you can see the general senses include stimuli like a touch, pain, or temperature. And then the receptors are located on the sensory neurons themselves. Okay, and then of course remember the dorsal root ganglion is where the cell bodies of all sensory neurons are located. The process that comes out of the cell body is called the peripheral process because it goes out to the periphery. You know, these receptor endings of these sensory neurons, they're coming somewhere, like say maybe in the skin or somewhere else in the body. And then of course the axons of the neurons of the central process take that information of the stimulus into the spinal cord via the dorsal root of the spinal cord, and then go into the cell body into the gray matter of the spinal cord. So your special senses detect very specific stimuli, while your general sensory receptors involve the detection of stimuli like pain, touch, and temperature. So, and many of your special sensors rely on receptors that are not neurons, while your general senses receptors are the receptive ends of sensory neurons. Also, special sensory organs are generally confined to the head. So all information then will travel to the axons of the different cranial nerves, while your general sensory information travels on the axons of both cranial and spinal nerves. So those are some of the basic differences. And now let's look at the process of smell, olfaction. The olfactory system allows us to detect odorants, which are chemicals. And then that chemical stimulus then is converted into an electrical signal that then can be interpreted by the brain. The brain can only read information that's an electrical. So as you can see, your olfactory bulb at the very top of the image will contain axons of olfactory nerves. Okay, and then you have olfactory epithelium in your nasal cavity. Okay, which are bipolar neurons, as you can see from figure B. Okay, and the olfactory cilia are the receptive ends that are basically hanging into the nasal cavity that would take that odorant, which is a chemical, through that bipolar neuron, and then it takes that message and then sends it eventually to the olfactory bulb where the mitral cells are located, and then the stimulus signal will then go into the axons of the olfactory nerve. So you have, so in your olfactory pathway, you have 3 types of cells in the olfactory epithelium. It's basically specialized cells located in the roof of the nasal vestibule, where you see that olfactory epithelium in figure A. And those specialized cells have 3 cell types: olfactory neurons, which are your olfactory cells. Basal cells are cells also located here next to the olfactory cells, olfactory neurons. The basal cells are stem cells that can produce new olfactory neurons when needed. And then you have your supportive cells. You can see the supportive cells in your picture at the bottom of figure B. Okay. Those are all located in the olfactory epithelium, and the basal cells only have a lifetime of about 7 to 10 days. But they will replace these olfactory neurons as they become damaged. They can only live for about 60 to 90 days. So here's the location of your olfactory epithelium in your nasal cavity. This shows you the cribriform plate of the ethmoid bone, the middle and inferior nasal concha, and below those are the nasal meatuses. So this is the inhaled air containing the odorants, which are aromatic chemicals, and that would be the stimuli, okay? And then they basically reach the olfactory epithelium, where those odorants are then transduced into a signal called an action potential, which will eventually reach the olfactory bulb. And this is a close-up showing you the structure of the olfactory epithelium, showing you the basal cells, the stem cells, which are the cells that replace the olfactory neurons. And then you have the supportive cells, which are anchoring cells. Okay, then you have the cribriform plate, which is where those axons of those olfactory neurons pass through. That's the reason they have so many perforations in that cribriform plate. And then they synapse with the mitral cells in the olfactory bulb. And those axons of the olfactory bulb then lead and become the olfactory nerve, cranial nerve number 1, which lies just below the frontal lobe of the brain. This is an image showing you the olfactory cilia with these dendrites that receive the signals, and many olfactory cilia. Remember, these cilia will increase the surface absorptive area. Okay, let's look at how the physiology of olfaction works. Okay, remember, odorants in the inhaled air are first detected by the olfactory neurons. Okay, and those chemical stimuli are then transduced into an electrical signal, which are then transmitted to various regions of the brain to be identified. So first, you have the binding of the odorant to its receptor that activates the G protein. The activated G protein, remember the stimulus, I remember, was the odorant that was bound to this receptor site. So once that activated G protein is triggered, it triggers the enzyme called adenylate cyclase, which is this little blue ovoid figure in step 2, and that converts ATP to a messenger called cyclic AMP. Cyclic AMP then opens up the ion channels. That allows your sodium and calcium to enter the cell. And that will then cause depolarization and eventually generation of an action potential. And if threshold is reached, once that action potential is generated, then your transduction has completed, has occurred. Okay. So remember, your primary olfactory pathway neural signals will then carry that olfactory information to the different regions of the brain for detection and identification of the different odors. Your olfactory tract is, though, the only pathway that does bypass the thalamus. That is a test question. Okay, so remember that. What is the primary olfactory cortex responsible for? It's primarily responsible for awareness and identification of odors. I think that's another test question. The primary olfactory cortex is primarily responsible for awareness and identification of odor. Okay? The amygdala, hippocampus, hypothalamus, and your components of your limbic system. These all receive information from your primary olfactory cortex for smell. It also allows us to evoke an emotional and visceral response to odors. Okay. Now let's look at the gustatory pathway, taste, which is in this figure 15.4. This shows you the anatomy of the tongue and the taste buds. Remember, gustation or taste involves chemoreceptors also, just like smell. And they're also stimulated by various chemicals. Taste begins with stimulation of these specialized receptor cells called taste buds. And that's what you see in this image here. The tongue is covered with taste buds, little small clusters of receptor cells and supportive cells scattered about the tongue and the other surfaces, especially in the posterior oral cavity, all the way into the esophagus. Taste buds are scattered and covered all over the tongue and in the oral cavity. They have these little rounded projections called papillae. And these papillae are classified based on their shapes. Valate or circumvallate, these are on the back of the tongue. These are fairly large. You can see a valate papillae in figure B from this longitudinal view here. They're on the lateral, the taste buds are on the lateral surface of the papillae. Okay. But you have four types of taste buds, okay, and four types of papillae, okay. You have 5 tastes, but you have 4 types of papilla, okay. You have your valate papillae, fungiform papillae, folia papillae, which is basically on the lateral side of the tongues. You also have filiform papillae, which they don't show here in this image. Filiform papillae will be devoid of taste buds, though. Okay, they're basically for tasting texture of different foods. So this is just the generalized structure of a taste bud found within those papillae. Okay, figure B shows a longitudinal view of a valate papilla. And figure C shows you a taste bud within a papilla. Okay, these are the gustatory cells at the top of the figure C. You can see the gustatory cells in the micrograph and the illustration, those orange colored cells with the central nucleus. Then you have the supportive cells, which anchor the gustatory cells. And then you have the basal cells, those smaller blue colored cells that are your stem cells for producing new gustatory cells. And then you have these little pores at the very tip of the taste bud. And that, of course, is where the chemical, the different chemicals that produce our taste reach the taste buds. So this is an anterior view of the tongue, revealing the valate papilla on the back of the tongue, the foliate papilla on the lateral surface of the tongue. Now, foliate papilla will have taste buds in childhood, but as you reach adulthood, you lose the taste function, okay, of the foliate papillae, okay. Of course, the valate papilla will have hundreds of taste buds, and then the fungiform papilla located at the very tip of the tongue and on the dorsal side of the tongue, they'll be have numerous taste buds also. Notice your palatine tonsils in the back of your oral cavity and your epiglottis in the middle. So this is a longitudinal view of a valate papilla, showing you where they're located on the lateral surface. That's where you see the taste buds of the papillae. These are the sensory neurons, okay, and these would be the actual taste buds with the pores exiting the surface, okay, of the taste bud. Of course, these are scattered throughout the posterior side of the dorsal side of the tongue. And this is an actual picture of a taste bud with the supportive cells and stem cells and taste pores at the tip. So let's look at the physiology of taste. Okay, there's basically 5 tastes. They're sweet, sour, umami, bitter. Okay. So that's what we refer to as the gustatory or taste pathway. So let's look at the physiology of gustation. The sense of taste, remember, involves signal transduction of chemicals in food that are received and then changed into an electrical signal that then can be sent to the brain. So first you have a change in your ion movements that depolarize the the gustatory cell's plasma membrane. Then you get depolarization of the membrane, which opens up for voltage-gated calcium channels. Calcium enters the cell. That triggers the release of neurotransmitters, which then produce an action potential in the axons of your sensory neurons. And that can now be sent to the brain, eventually to its receptors in their appropriate cortex. Taste will go to the parietal lobe and the insula. These are the receptive endings of the sensory neurons in that figure 15.5. So, and this is your gustatory pathway, okay? It shows you first the axons. There's 3 nerves that carry taste, okay? Number 10, the vagus. Number 9, the glossopharyngeal. It innervates the posterior third of the tongue. And the facial nerve innervates taste on the anterior 2/3 of the tongue. Okay, this is, remember, that's where your fungiform papillae are located. So axons of your facial, glossopharyngeal, and vagus, 7, 9, and 10, will carry the taste stimuli from the tongue into the central nervous system, which is the medulla. The axons of those 3 nerves then terminate in the solitary nucleus in the medulla oblongata. Then synapses on those central sensory neurons, then they interact with the second order neuron, which takes the impulse from the solitary nucleus to synapse in neurons in the thalamus. Okay, and then they send the signal to the primary gustatory cortex in the parietal lobe. You also have information of taste that also goes to the insula, but that will serve the internal organs. Okay. This next view is basically the picture of the external anatomy of the eye. Of course, the eyes are protected by your eyebrows. That prevents, your sun's glare, prevents objects. Of course, you have your eyelids, which are protective. These have very sensitive receptors that allow you to blink when one is faced with an object that threatens to enter the eye. The medial angle where the eyelids, you got your upper and lower eyelids meet, that's called the medial commissure. And within that medial commissure is a little structure called the lacrimal caruncle. And these will be associated with our tear production later on by a gland called the lacrimal glands. The other side of the eye is called the lateral commissure. And then you got your eyelids attached to your eyelids. The eyelids are also called the palpebra, and the muscles in the eyelids will contain the palpebral muscles with these plates and glands called tarsal plates and tarsal glands. And then you can see these tarsal glands and tarsal plates within that palpebra, which is your eyelid, your upper eyelid and lower eyelid. And they will be on the upper region of the eye. Okay. On figure V is a lateral view showing you, of course, the eyebrows at the top. There's the levator palpebra superioris muscle, which elevates your upper eyelid. And then the tarsal plates and tarsal glands. What do tarsal glands do? They secrete a kind of a lipid-like secretion, a watery secretion that is produced when you sleep, and it prevents your eyelids from adhering to each other. Okay, so like when you get up in the morning, you get the sleep out of your eyes, what you're doing is removing the secretions from your tarsal glands. That's what we refer to as the sleep out of our eyes sometimes. And then you have these thin epithelial membranes called palpebral conjunctival membranes and ocular conjunctival membranes. The orbicularis oculi muscle, this is a muscle, of course, that allows you to close your eyelids and your eyes. And then you have 6 extrinsic eye muscles that are attached to the outer covering of the eye called the sclera. What you see here is the lateral rectus, superior rectus, inferior rectus. You don't see the inferior oblique and superior oblique on this though, okay? So here's a close-up showing you the external anatomy again of the eye, showing you the medial and lateral commissures, the eyelids or a palpebra, superior palpebra and inferior palpebra, and the eyebrows. And on a lateral view of the eyes, you can see the conjunctival membranes that cover the outer fibrous layer of the eye. That's called these transparent epithelial membranes. One's called the bulbar ocular conjunctival membrane, and the other one is called the palpebral conjunctival membrane. What happens, it folds in on itself and becomes a palpebral conjunctival membrane when it enters the eyelids. But the palpebral conjunctiva and the ocular conjunctiva are continuous. Okay, but they contain blood vessels. They're transparent. When you get an infection of the eye, like conjunctivitis, these are the transparent epithelial coverings that are involved. Okay. Of course, the eyelashes are for protection. Eyelids, blinking is for protection. And the extrinsic eye muscles, those that are attached to the sclera, are involved in eye movement. You have a special apparatus in the eye called the lacrimal gland. It's located in the superior lateral wall of the eye orbit, next to the fibrous tunic of the eye, where the sclera is. Okay, and it's going to produce tears, and tears are going to be produced by the lacrimal gland. Then they're ejected out of the lacrimal ducts. Okay, and then the tears are basically going to bathe the ocular membranes, those conjunctival membranes, and then they'll wash the eyes, and they'll eventually drain onto the medial commissure, and then enter that area called the lacrimal punctum, okay, and lacrimal sac. Lacrimal sac then leads to the nasolacrimal duct, which travels through the ethmoid and maxillary bones, and your tears will eventually enter through the inferior nasal meatus, bordered by the inferior nasal concha. This nasolacrimal duct is shown on the X-ray in Figure B, where you can see that lacrimal ductwork as it travels through the ethmoid bone and the maxillary bone, eventually entering the nasal cavity and the inferior nasal meatus. Okay. This is a close-up showing you the lacrimal glands, the lacrimal punctum, the lacrimal canaliculi that take the tears into the nasolacrimal duct, the lacrimal sac where the tears are stored, and the nasolacrimal duct, which again travels through the ethmoid and maxillary bones to be transported into the nasal cavity at the inferior nasal meatus, eventually out your nostrils. And there's an X-ray of lacrimal ductwork with a radiographic opaque dye, so that a lot of times these ducts get obstructed. The person can't produce tears, which are protective, and they have to sometimes have it cleared. But that just shows you the lacrimal sac, the lacrimal canaliculi, the ducts that take the tears, then they eventually into the inferior nasal meatus. So that should be clear all the way up. And now we're going to look at the interior of the eyeball. There's 3 layers to the eyeball. The outermost layer is your fibrous layer. And it consists mostly of this white collagenous connective tissue called the sclera. The sclera covers most of the eye, the posterior 4/5. The anterior fifth of the fibrous layer is called the cornea. The middle layer of the eye is called the vascular layer. It consists of the posterior choroid, which is kind of colored red-orange. Okay, and anterior, the choroid is continuous with a structure called the ciliary body and ciliary muscle. And then continuing, it becomes the iris, which the iris contains these pigmented epithelium, but it also contains these 2 radial muscles called the pupillary constrictor sphincter and pupillary dilator muscles. And these are going to be activated by light energy and your autonomic nervous system, as we'll see later. Okay, but anyway, that's all part of your vascular tunic. The choroid, ciliary body, iris. Okay, those are all part of that vascular tunic, that middle layer. Okay, and then the innermost layer is the retina, the sensory layer. The retina layer will contain our photoreceptor cells that receive light energy, and eventually that's what eventually will be processed to an image. So it's directly responsible for our vision. Another feature that you see on this view is the optic nerve exiting the eyeball. Notice that the optic nerve, where it exits, that area, there's no photoreceptor cells. rods and cones. And that's what we call our blind spot. So our optic disc is what produces our blind spot because there's no retinal photoreceptor cells located there. Just to the lateral side of the optic disc is an area called the macula lutea. And right in the very center of the macula lutea is a concentration of cones called the fovea centralis. This area is responsible for our acute vision, our ability to see fine detail. If you've ever heard the term macular degeneration, this is the area that deteriorates. Very important area because this is where our ability to see clearly occurs. Okay, so anyway, I just kind of wanted to show you what some of these structures are. The central artery and vein of the retina, they course in the middle of the optic nerve. Okay, the ora serrata, this is this irregular margin. Okay, and this is going to meet the edge of the. This is basically where the choroid and the retina meet. Okay. Okay. Now looking at the suspensory ligaments, these are ligaments that attach the ciliary body with the lens. The lens is also a transparent structure that contains a protein called crystalline protein, and it's attached to the ciliary body by these ligaments called suspensory ligaments. Okay. So the layers of the eyeball are the fibrous layer, which consists of the sclera and the anterior cornea. Cornea, of course, is transparent because the collagen fibers are arranged at parallel, and that allows light to enter the cornea. Cornea should be equal thickness all the way around. Okay, and we'll see what happens when it's not equal thickness later when we look at the conditions of the eye. Again, fovea centralis, this is the area where your concentration of cones is at its greatest, so that's what's responsible for our acute vision. So the iris, ciliary body, choroid, these are all part of your vascular layer. The retina is part of your sensory layer, and then the fibrous layer is the sclera and the cornea. The lens, what the lens does, it divides the eye into two cavities, an anterior cavity in front of the lens, a posterior cavity behind the lens. And in the anterior cavity, anterior to the lens, you have two chambers, a posterior chamber and an anterior chamber. And within that, those two chambers, the ciliary body will produce a fluid called aqueous humor. And aqueous humor will flow from the posterior chamber in through the pupil into the anterior chamber. And we'll talk about glaucoma later when we see what happens when aqueous humor accumulates in higher than normal amounts, and it increases the pressure within the eye. And that can cause a condition called glaucoma. So here's another magnified view showing you the fibrous layer with its sclera and cornea, and the vascular layer with the ciliary body and muscle and choroid. And there's that ora serrata again. Okay, and there's just another magnified view showing you the layers of the eyeball. So what happens when you shine light into the eye? Well, the picture in the middle shows you what happens in normal vision. Okay, when you have normal, when you don't have, when it's just of average size. Okay, the pupil can be gauged with these. If you go into the healthcare field, you will get these little cards. You can see these little dots, and they'll show you the size of the pupil and what it should be. Okay, so if you increase your light intensity, notice that pupil will constrict in size. Okay, or if the parasympathetic system is activated, that will also cause the pupil to constrict. Okay, so the autonomic nervous system can regulate the size of the pupil. Whereas when you decrease light intensity, like when you go from a light situation to a room in the dark, that will cause your pupil to dilate. Okay, and that's a responsibility of one of those muscles in your iris called the pupillary dilator muscle. When you constrict your pupil, the pupillary constrictor muscle will contract, and that will cause the pupil to constrict. Okay, the pupillary constrictor is on the inside, and the pupillary dilator muscle is on the outside. So when you decrease light intensity, go to the dark, the pupillary dilator muscle will be activated, and that will dilate your pupil to allow more light to enter. So pupillary dilator muscle contracts to make the pupil larger. Pupillary sphincter muscle contracts to make the pupil smaller. And these are both present in the iris. Of course, you also have pigmented epithelium in the iris, which produces melanin, and that produces the color of your eyes. The more melanin you have, the more, and of course, that's going to also contribute to the color of your eye. Also, I might just mention briefly that whenever you shine a light into, let's say, the left eye or the right eye, the same thing will happen to the other eye. Let's say when I shine a flashlight into, let's say, the left eye, okay, the pupil will instantaneously constrict. At the same time, the pupil on the right will at the same time constrict. And that's called the consensual pupillary response, and that's because you have fibers not only from the optic nerve, but also from the oculomotor nerve that regulates the pupil size, okay, the pupillary constrictor pupil from the oculomotor nerve, corner number 3, on both sides. And what that does, that's a protective measure just in case you're exposed to increased sunlight, let's say blinding glare from the sun, or some sort of bright light threatens can threaten your integrity of your eye structures, and so both pupils will constrict, basically protect you from that increased blinding light that could potentially be damaging to your retina, especially if you directly looked at an eclipse or the sun. Now, what you see here in this image is what an ophthalmologist sees or an optometrist sees when he looks in the back of your eye when he's doing a visual exam. This is called a fundus exam. What you see when you take an instrument called an ophthalmoscope, you see the optic nerve exiting at the optic disc, and that produces this little yellow spot here. You also see these little branches coming off the central retinal artery. Okay, these are retinal blood vessels, and just lateral to the optic disc, where the optic nerve exits, remember that's where your blind spot is produced, you have an area called the macula lutea, and at the very center of the macula lutea is the focus centralis, where your acute vision and where you see only cones, where your color vision and your acute vision is located. That's called a fundus, and this is the back of your eye, and that's what you would see if you looked at the back of your eye with an ophthalmoscope. So fovea centralis, macula lutea, optic disc where the optic nerve exits, and branches of the central retinal artery. Of course, that looks like a healthy retina. If you were looking at a diabetic patient, you possibly could see a greater number of these retinal blood vessels. And that's called diabetic retinopathy. Okay, and the doctor has to come in and use a laser and basically cauterize these excess blood vessels, so that because these blood vessels get so numerous that they have to get rid of a lot of those blood vessels so the person can see. That's called diabetic retinopathy, and it occurs especially in diabetics. If you want to find your blind spot, remember your blind spot is produced because your optic nerve, when it exits, there's no photoreceptor cells. If you want to look for your blind spot, and if you want to test your left eye, you cover your right eye with your hand or a paper or a patch, and then while you're looking at the X, okay, covering your right eye with your left eye and concentrating on the X with your left eye open, then you move your book or whatever paper that you have this image on, and you bring that closer to your eye, okay? And at that point, where the circle disappears, as you bring that book closer and closer to your eye, that will be your blind spot. Most people have a blind spot about 8 to 12 inches in front of their eyes. This picture on the bottom shows you how to fill in your blind spot. What you do here is, if to test your left eye, you cover your right eye and also again focus on the X. You slowly move the book toward you, okay? And you'll move it closer and closer to you until, as you're only concentrating on the X, okay? You move it close enough to you until you see that the pencil on the left here appears intact and there's no gap between the two parts. That would be your blind spot. And again, it should be about 12 inches in front of your eyes. So when that gap in the pencil hits your blind spot, those areas in your brain then will fill in the missing information so that the pencil appears intact. And then if you test your right eye, if you want to test your right eye, you turn your book upside down and then cover your left eye. And you repeat the process. So to test your left eye, cover your right eye, focus on the X, slowly move your book toward you until the dot disappears. To test your right eye, you cover your left eye, and you focus on the dot, move the book until the X disappears. Same thing here. You're filling in your blind spot with this exercise. Test your left eye, you cover your right eye, and you focus on the X. You slowly move your book toward you until the pencil appears intact. When the gap in the pencil hits your blind spot, those areas in your brain will then fill in the missing information and the pencil will appear intact. You do the same thing to test your right eye. Turn your book over, upside down, cover your left eye, and repeat the process. Okay, some of these other features of the eye that you see here. You have two chambers to the anterior cavity. Remember, the anterior cavity is the part of the eye that's filled with aqueous humor anterior to the lens. Okay, posterior cavity is behind the lens, and it contains a gel-like material called vitreous humor, and it's located in your posterior cavity. The vitreous humor is a little more viscous than your aqueous humor is. Okay, the vitreous humor serves to keep the retina firmly attached to the vascular layer. Okay, and it provides shape to the eyeball. Okay, and it does minimally refract the light rays, as well as the aqueous humor refracts the light rays. Okay, the anterior cavity is anterior to the lens. The anterior cavity consists of a posterior chamber and an anterior chamber. The ciliary body will make a fluid called aqueous humor. Less viscous than vitreous humor. It's more watery. It has a consistency closer to what plasma is. Aqueous humor is secreted by the ciliary body, and then, as it's secreted, it then circulates from the posterior chamber, goes through the pupil, as you see those white arrows, and then goes through the pupil into the anterior chamber of the anterior cavity. And then that fluid will then be drained into a sinus called the sclerovenous sinus, or the canal of Schlemm. Okay. And when people have too much aqueous humor and it's not drained properly by the sclerovenous sinus, also called the canal of Schlemm, S-C-H-L-E-M-N, the pressure within the eye, they call the aqueous humor, will start building up. And that increased pressure can put pressure on the vitreous humor back here, or the vitreous body back here, and that puts pressure on the retina and the optic nerve. And that's what we refer to as glaucoma. It's the second leading cause of blindness. Macular degeneration is the first. Okay, this is just showing you the visible light spectrum, where it lies in the electromagnetic spectrum. As you can see, your visible light ranges between 350 to 750 nanometers. Your larger wavelengths are your radio waves and microwaves and infrared. Your smaller wavelengths are your ultraviolet, X-rays, and gamma rays. So yellow would have a wavelength close to about 600. Red, orange, about 660. Deep red is about 750. Your shorter wavelengths are closer to your blues. 350 would be purples are your shortest wavelengths of light. Middle is about 500. Okay, this is about 500 nanometers is about where rods focus. Rods are the top photoreceptor cells in your retina, but they only detect one wavelength, whereas the other photoreceptor cells called cones, they can detect 3 wavelengths. They detect red, blue, and green. But that's what we call our visible light spectrum. Ranges between 350 to 750 nanometers. Also, light rays bend as they strike a surface. Distant objects, the light rays are almost parallel, but as the object gets closer, it bends at a greater angle. Okay, the medium that the light rays travel will also have the ability to bend the light rays. Air has what we call a refractive index of 1.0. That means light rays are going to be straight on. Okay, they're going to be linear. So density is similar to air. The light will not refract. Okay, whereas water has a refractive index greater than 1. So as when light rays enter the water surface, when they enter that water surface, that the light ray will bend. You can see that demonstrated if you've ever taken a pole, a long pole, you know, where they have those nets where you clean out your pools, and you put you submerge the pole into the swimming pool water, and you can see the pole bend as it goes through the water, and that's because the water has a refractive index greater than 1.0 than what air is. So the density of the medium will refract the light rays. So density similar to air, light will not refract, so it's basically a parallel line, a singular line. In water, when light goes from air to water, water has a refractive index greater than 1, so that causes light rays to bend, as you can see from the demonstration. Also, the lenses are what opticians use to, and optometrists use to correct errors in vision. Okay, we're going to talk about nearsightedness and farsightedness, myopia and hyperopia also later. But a convex lens is one that converges light. In other words, light rays as they enter, they're basically, you know, if they're looking at a distant object, they'll be almost parallel. Well, a convex lens, which is thicker in the middle than the edges, will converge or bring all the light rays to a focal point. Okay. Whereas on the other hand, a concave lens is thicker on the edges but thinner in the middle. So when light rays enter a convex surface, okay, the light rays will diverge, spread out, so they'll be unfocused. Okay. But we'll use these types of lenses, concave and convex, to refract the light rays with individuals that have errors of refraction. So if you look at this illustration, as your distances are less than 20 feet, the light rays will diverge sharply. Okay, whereas the distance is greater than 20 feet, you can see those light rays are almost parallel. The more parallel the light rays, the less your ciliary body has to work to bend the light rays. And that's the reason it's important to look at distant objects when you're driving to relax your eyes. Whereas when you're reading a book, you're constantly contracting your ciliary body because you're trying to bend those light rays so that you can see the words on your book. Okay, and that puts strain on your ciliary body and muscle and gives you eye strain. Okay, so if you see a person that has normal vision, okay, emmetropia, that means the lens is flattened for distant vision. If you look at an apple on a distant tree, like you see in this situation, figure B, the rays are almost parallel when they reach the eye, as you can see from the illustration. So the cornea and a flattened lens is all that's needed to provide enough refraction to focus the light on the retina and get a sharp focal point. On the other hand, if you bring an apple, let's say, closer to your eyeball, to your cornea, the rays are diverging, as you can see, when they reach your eye. Okay, now you don't need this flat lens, but you need a more rounded lens. Okay, because they will bend the light rays and converge them, because now it's become convex in shape. And that will bend those light rays so that those light rays can rest on the focal point in the retina. So a rounded lens will provide the additional refraction that's needed to focus light rays on the retina. What you're doing is you're contracting the ciliary body, which lets off the tension on these ligaments, causes the ligaments to slacken, and then when the suspension ligaments slacken, then the lens will become more rounded in shape. It rounds up. So this is at distant images that are seen at distances greater than 20 feet. The light rays are parallel. At less than 20 feet, the light rays will diverge greatly. On the other hand, if you look at an apple at a distant tree, the rays are almost parallel when they reach the eye. So the cornea and a flattened lens will bring the image to a sharp focal point to focus the light on the retina. And if you bring, let's say, an apple in your hand, the rays are diverging when they reach the eye. So now you need a more rounded lens, and that will provide the additional refraction needed to focus the rays on your retina. So what is happening here is your ciliary body moves away from the lens, putting tension on the suspensory ligaments and flattening the lens when you're looking at a distant object, because the ciliary muscle, the ciliary body is relaxing. And then when you look at something very near, which is, by the way, the changing of the shape of the lens to view near object, that's called accommodation. So what happens in accommodation is when you're looking at, let's say, that apple that's near you, the smooth muscle of the ciliary body contracts. That takes the tension off the suspensory ligaments. Okay. Now that contracted ciliary body moves closer to the lens, removing tension on the suspensory ligaments. They start to slacken, and that allows the lens to resume its more rounded shape. It rounds up. So two of the common conditions that occur are hyperopia or farsightedness. You can see distant objects but not near objects. That's when the eyeball is too short. Okay. Whereas in myopia or nearsightedness, the eyeball is too long. Okay, so your focal point is going to rest in front of your retina. So in hyperopia, the figure A, farsightedness, the eyeball is too short, so the lens cannot round up enough, and therefore light rays focus behind the retina when viewing near objects. So you correct this kind of refractive error by giving them a convex lens, which converges the light rays before they reach the eye. And therefore, the lens can now provide the rest of the needed refraction to get a clear focal point so the person can see and focus. On the other hand, in myopia, where the eyeball is too long, the distance between the cornea and the lens is too great. So now, uncorrected, the lens cannot flatten enough and the light rays focus in front of the retina because the retina is back here, it's too long. So you give this type of individual with myopia a concave lens, okay? And that will basically diverge the light rays, spread the light rays apart, okay, before they reach the eye. So that now you need minimum refraction of the lens. That's all that's necessary to focus the rays on the retina. So hyperopia, the eyeball's too short. So you bring the focal point up to their retina and you use a convex lens. And that will be enough to converge these light rays so that the light rays before they reach the eye, so that now the lens can provide the rest of the needed refraction. In myopia, on the other hand, the eyeball is too long, so now their focal point is in front of their retina, uncorrected, so they've got a blurred vision. Give them a concave lens, and that diverges, spreads apart the light rays before they reach the eye. So you need minimum refraction now of the lens, and that's enough to focus the light rays onto the retina. So now you can see clearly. This is an image of the retina. This is a micrograph on the retina in figure B, and an illustration of the retina in figure A. You can see the choroid in figure B in this micrograph, and in figure A you can see the choroid. That's on the far right of both pictures. Okay, and then just below the choroid, just deep to it, remember we're basically going towards the vitreous body, okay, the interior of the eyeball. So this is a pigmented layer of the retina. The pigmented retina is basically a backing to the retina. It prevents scattering of light. It keeps the light focused. Okay, and that's the reason it's pigmented. Okay, the next layer that you see are your rods and cones. Okay, Your cones are cone-shaped, and your rods are long cylindrical shaped. And they contain chemicals called rhodopsin and photopsin. Rhodopsin will react to one wavelength, photopsin several wavelengths. So rhodopsin is in your rods, and photopsin is in your cones. But anyway, these rods and cones were responsible. These are the receptors for the photons of your light energy. They're going to produce the image. And then these are the nuclei of the rods and cones. You also have a lot of horizontal cells. Okay, these are the bipolar cells. Bipolar cells are basically relay cells. They take the message. Okay, and then you have the ganglion cell layer. Okay, and then the axons of the ganglion will eventually become the optic nerve. So those are your layers of your retina. You got two layers up to your retina: the neuroretina and then the pigmented retina. And then your choroid is your middle layer of your eyeball. Remember, the retina is part of the sensory layer. Choroid is part of your vascular layer. So here's another image showing you the retinal ganglion cells, the bipolar cells, the rods and the cones, the pigmented retina, and then the choroid. Choroid is kind of like the backing of a mirror. Okay, it's like the lead backing to a mirror. It basically prevents any light rays from scattering and keep it all contained within the neuroretina. This is just another image, another micrograph of the retina showing you the choroid, the pigmented retina, the rods and cones, bipolar cells, and the retinal ganglion cells. Okay, here's an image showing you the cones and rods again. This shows you the structure of a cone. There's the pigmented retina. So these are the outer segments of the cones, which contain the chemicals photopsin. There's the inner segment, the cell body. Okay, and these are the synaptic terminals. Okay, and then the rods have these thousands of rod discs, which contain rhodopsin. Okay, they're layered on top of each other. There's the mitochondria and the inner segment, and then the cell bodies. And this figure B is rhodopsin in the disc membranes of a rod. And this is what is going to react to the light energy when it enters the eye. It will be partly responsible for producing an action potential. Okay, and then the absorption of the various wavelengths of light by the cones and rods. You can see that your blue cones are about 420. Your green cones are about around 530. Your rods respond to about 500 nanometers. And the very tip, the bright red, would be around 700. But these are the different wavelengths that the different cones, red, blue, and green, and rods respond to, reflect. And that's, again, another structure of a cone on the bottom and a rod at the top. And this is your rhodopsin in your disc membrane of a rod. There's your rods and cones again. And there's your visible light spectrum, showing you the light absorption as a percent of maximum and the wavelength on the bottom. It ranges between 350 up to about 750 nanometers of light. Remember, this is the very middle of your electromagnetic spectrum. Remember, microwaves, radio waves, those would be larger waves. X-rays, gamma rays, infrared would be less. Ultraviolet light, gamma rays, X-rays, those would be much smaller, whereas infrared, microwaves, radio waves, they would be larger. So, but that's the visible light spectrum is the very middle of your electromagnetic radiation. So let's look at how we see. Okay, in the light, your photoreceptor cells will hyperpolarize. So light causes the retinal, that chemical in your disc, okay, to separate from the opsin. Transducin and PDE are then activated. that causes the sodium channels to close, and the photoreceptor cell hyperpolarizes. That's what you see in light. Okay. Now in the dark, the opsin and the cis-retinal combine to form rhodopsin in the disc membrane. Then the sodium will now enter the outer segment of your photoreceptor cell and depolarize it. Okay. So in this case, you're not going to see an image during depolarization. In the bottom image, you have hyperpolarization because of the closing of the sodium channels, and that's what occurs in light. And that's kind of the opposite of what you see in the action potentials in your other general senses. So first, the opsin and the cis-retinal combine to form rhodopsin in the disc membrane in the dark. Second, the sodium enters the outer segment. The photoreceptor then depolarizes. Light will then, the light will cause the retinal to separate from opsin, like you see in the image. Transducin and GMPD are then activated. That causes the sodium channels to close, and now your photoreceptor hyperpolarizes. Okay, that's what occurs in light. So what you have in the dark, the retinal ganglion cells are not stimulated because the retinal ganglion cell does not produce an action potential, because you got this glutamate inhibiting these bipolar cells and reducing its release of neurotransmitter. No neurotransmitter, you don't get an action potential. Okay, but it occurs with depolarization and releasing that glutamate. Whereas in light, in conditions of light energy, light will hyperpolarize your photoreceptor. Now it stops releasing glutamate. Bipolar cell now is freed from inhibition and it depolarizes. That releases the neurotransmitter into the retinal ganglion cell. The retinal ganglion cell then produces the action potential, and that's sent to the brain via the optic nerve. So again, in the dark, the retinal ganglion cells are not stimulated, mainly because the photoreceptor depolarizes and releases glutamate. Glutamate then inhibits the bipolar cell and reduces the neurotransmitter. Therefore, you don't get an action potential in the retinal ganglion cell. So you don't see an image in the dark. On the other hand, in the light hyperpolarizes the photoreceptor cell, stops releasing glutamate. Bipolar cell is now freed from inhibition and depolarizes. That releases neurotransmitter onto the retinal ganglion cell. The retinal ganglion cell then produces an action potential that's then sent to the brain via the optic nerve. So, on your visual pathway, okay, your right and left eyes are in front of our heads. Okay, so that's what that's going to do. It's going to give us a visual fields where both eyes, in binocular vision, you can see both visual fields being represented, where the right eye only, in binocular vision, can only see this part of the visible spectrum, this red area. And on the left side, you can see the left eye only, and this is also called monocular vision, one eye vision. You only see the left visual field, that in deep blue. So the retina of each eye will detect the visual stimuli from portions of the right and left visual fields, and they're represented by the red and the blue color codes that were used in the visual field representation. So the temporal retina from the right eye is taking visual information from your left visual field, okay? And your nasal retina of your right eye is taking visual image from your right visual field, okay? And then those fibers run via the optic nerve. Some visual stimuli will then cross the optic chiasma, mainly the nasal retina of the right eye. So that some visual stimuli will cross at the optic chiasma, so that all stimuli from the right visual field are going to be processed by the left hemisphere. That's the reason you have these red optic radiations on the right side here of the visual cortex. And the same thing for the left side of the visual cortex in the occipital lobe. It's going to be representing the left visual field, okay, as you can see from the colors. Okay, so some visual stimuli cross the optic chiasma, so that all stimuli from the right visual field are processed by the left hemisphere, And stimuli from the left visual field over here will be represented by the right hemisphere. And eventually the visual stimuli will then travel from the thalamus. And they also have a relay that goes to the superior colliculus of the midbrain. Okay. Well, anyway, the visual stimuli will then travel from the thalamus, as you can see, by the optic radiations to the primary visual cortex in the medial part of the temporal lobe. So here it is again. Okay, so the retina of each eye will detect visual stimuli from portions of both right and left visual fields, as you can see by the red and blue colors. Okay. Second, the retina of each eye detects the visual stimuli from portions of the right and left visual fields. Some visual stimuli will cross over, particularly when we're looking at the right eye. The nasal retina of the right eye will cross over at the optic chiasma to the left side, to the right side. The nasal visual field of the right eye will cross over to the left hemisphere. So you have some visual stimuli crossing at the optic chiasma, so that all stimuli from the right visual field over here on the right side are processed by the left hemisphere. And the stimuli from the left visual field on the left side of the eye are going to be represented by the right hemisphere. That's when you have the blue optic radiations going to the occipital lobe, colored blue, because they're going to represent that area in the left visual field. So if you look at this big picture, the cornea and a flattened lens, what you see is the left visual field, the surfboard, the right visual field, the surfer. Okay. So first, the cornea and a flattened lens provide enough refraction to focus light rays on the retina, because that's out in the distance. He's probably on the beach. You're out on the beach looking at this man on a surfboard fall off. Okay. So you don't need much flattening of the lens because the subject is more than 20 feet away. That's distant vision. Now your photoreceptors hyperpolarize, the retinal ganglion cells and the expotentials through the optic nerve. They'll send the expotentials on through. Then you'll have selected axons that cross at the optic chiasma, so that stimuli from each half of the visual field, okay, are going to be sent to the opposite hemisphere. So eventually the primary visual cortex will provide a conscious awareness and an initial processing of shape, color, and movement. Okay, so each hemisphere will then process stimuli from the opposite eye's visual field. So when your cornea and a flattened lens provide enough refraction to focus light on the retina, because the subject is more than 20 feet, now your photoreceptors hyperpolarize, the retinal ganglion cells then send expotentials through the optic nerves. Then you get some selected axons crossing over at the optic chiasma, so that stimuli from each half of the visual field are then sent to the opposite half of the hemisphere, the opposite hemisphere. So your primary visual cortex then will provide conscious awareness and initial processing of shape, color, and movement. So each hemisphere will then process that stimuli from the opposite eye's visual field. Astigmatism and color blindness. Color blindness is basically when you have a defect in either the red or the green genes for producing color vision. Your color genes are on the X chromosome. Okay, so if you have color blindness, you would not be able to see this number. So hopefully you can all see this number. The number that's represented here is 74. But if you had color blindness, either for red or green, instead you would not see 74, you would see the number 21. Males typically will have more color blindness than females. It's 8 to 10% of males, less than 1% of females. That's because males have only one X chromosome. And since the color genes, red and green, are on the X chromosomes, that means that a male only has one X chromosome, so if he has a defective color gene, he's going to have color blindness usually. But if a female has a defective color gene on one of her X chromosomes, she has another X chromosome. which is normal, so she won't be affected by color blindness. She'll have normal color vision. The only way a female is going to get color blindness is if she is unlucky enough to inherit both defective color genes on both her X chromosomes, and that occurs in less than 1% of the population of females. Okay, I just want to briefly introduce hearing and balance. There's 3 layers to the ear: outer, middle, and inner. The outer ear consists of the auricle, the earlobe, and the external auditory canal. Okay. The tympanic membrane is the boundary between the outer ear and the middle ear. The middle ear contains what we call the bones of vibration, the malleus, incus, and stapes, or hammer, anvil, and stirrup. The stapes is on the other side of the oval window, which is the beginning of the inner ear. Okay, and then the inner ear contains the semicircular canals with their semicircular ducts inside within this bony labyrinth. Notice from the middle ear and even part of the external auditory canal, these are all encompassed in bone, okay, porous bone. But anyway, the cochlea is where the organ of hearing is located. Okay, that's where the organ of corti for receiving sound waves, frequencies of sound waves. Okay, and then the semicircular canals and vestibule, which contains the utricle and saccule, vestibule are responsible for what we call our static equilibrium. Okay, our position of our head in space. Whereas our semicircular canals, with their ducts inside, which carry a fluid. Okay, these are responsible for our moving, our dynamic equilibrium, like when we go around in a circle or go around in a merry-go-round. Okay. And in the cochlea, we'll have 3 hollow chambers, scala tympani, scala vestibule, and scala media. And they will also be filled with fluid. 2 of them will be filled with perilymph, and 1 will be filled with endolymph. But this is going to be responsible for bending of hair cells in a special organ called the organ of corti, and that's what responds to frequencies of sound waves. And then there's a tube called the pharyngeal tympanic tube that extends from your pharynx opening, in the back of your throat, to the middle ear. It's also called the auditory tube or eustachian tube. This tube is what equalizes the air pressure between the internal pressure of your body and the external pressure of the environment. It's the eustachian tube or pharyngeal tympanic tube, also called the auditory tube, that basically has problems with pressure changes, like when you go up in a higher altitude, okay, or if you go in a submarine to the bottom of the ocean floor. So there's the outer ear with its structures. The lobule is the earlobe, external auditory canal. Auricle is the pinna. That's this top. That's your earlobe, the pinna. This area that contains this cartilage that you see here, this elastic cartilage. Here's your middle ear with its ossicular bones, malleus, incus, and stapes. The inner ear with the semicircular canals with their ducts inside filled with fluids, perilymph. Vestibule with the utricle and saccule for our static equilibrium. By the way, each of these semicircular canals, anterior, posterior, they're arranged at right angles, so they record movement in each direction of the planes. The round window is the terminal ending of the cochlea. Okay. And the entry to the cochlea is the oval window on the other side of the stapes. You also have a muscle called the tensor tympani muscle, and another muscle called the stapedius muscle, which they don't show here. But they're going to serve to basically serve to stabilize the ossicular bones during very extreme sounds. Here's a healthy tympanic membrane on the very far left, viewed through the external auditory canal with an otoscope, which is a special instrument that can produce a bright light so that you can see inside the auditory canal. Tympanic membrane and acute otitis media, which is acute ear infection that spreads almost always from your oral cavity, usually from a tonsil like the adenoids or pharyngeal tonsils, even though the other tonsils can result in acute otitis media. Mainly occurs in children, primarily because children have an auditory tube, a pharyngeal tympanic tube, that's wider and more horizontal than what you see in adults. Okay, and as you get older, the auditory tube will narrow and and move at a right angle. When you have severe acute otitis media and as you start having a discharge, chronic otitis media, then sometimes they have to drain that fluid on the other side of the membrane. And that's when tubes have to be inserted into the tympanic membrane. That's called laryngotomy. So this is an illustration showing you the inner ear structures. The blue area, those are your semicircular ducts with the anterior, posterior, and lateral canals. The vestibule is the utricle and saccule. Okay, and then your oval window and round window. The oval window is just on the other side of the stapes. Round windows over here in the cochlea. The blue area in the cochlea, that spiral-shaped snail-like structure, is going to have three chambers. Okay, you can have perilymph in the chambers that surround the blue chamber, and that's the scala vestibule and scala tympani. Okay, and in the chamber in the middle, it's called the scala media, or the cochlear duct. And that's where your organ of corti, your organ of hearing, is located, which has these little hair cells that respond to different frequencies of sound waves. Now, the nerve that transmits the information for hearing and balance, hearing is via the cochlear nerve, which is a branch of the vestibulocochlear nerve, number 8. And the vestibular nerve goes to the organs of balance, equilibrium, the semicircular canals and the vestibule. Okay, so for both dynamic, moving equilibrium and static equilibrium, where you're going up and down an elevator, those are going to go into the vestibular branch of the vestibulocochlear nerve. Those are all part of the inner ear. So when we say neural deafness, we're referring to a problem with, the nerve or possibly the cochlea, the organ of corti. When we see conduction deafness, those are usually deficiencies either in the ossicular bones of vibration, the malleus, incus, or stapes. A lot of times the incus and the incus and the stapes are fused together and they don't vibrate, and those serve to amplify the intensity of the sound so that you can go from an air medium into a liquid medium. So here's a diagram showing you your three chambers within the cochlea. This is basically unwound. So you can see the scala vestibule contains perilymph, scala tympani contains perilymph, and then the scala media or cochlear duct contains endolymph. Endolymph is very similar to plasma. Okay, and you can see the hair cells within the spiral organ, also called the organ of corti. And this was where your frequencies are registered. The very bottom is a membrane called the basilar membrane, and then separating the cochlear duct with the scala vestibule is the vestibular membrane. But the basilar membrane is the very bottom of the organ of corti. And it's when the basilar membrane vibrates that also is influenced by the movement of these fluids within these two chambers, which are produced by the vibrations of air molecules through your sound frequencies. Sound frequencies, basically we have a hearing range of between 20 to 20,000 hertzes, or vibrations per second, or cycles per second. That's our range of hearing, 20 to 20,000. Our decibel, our loudness of sound, the other quality of sound, which then refers to the amplitude of the sound wave. When you start injuring these hair cells is when you get above 90 decibels of sound intensity. Most rock bands are between 90 and 100. A jet airplane is about 120 decibels. And that's the reason when people working in air landings, they have to wear those ear protection. There's your round window and oval window. The oval window is the entry to the cochlea. The round window is the exit. The modiolus is the bony inner structure, and these are the chambers within the cochlea. The scala vestibule, the top chamber, the scala tympani, the bottom, and the scala media in the middle. The scala media is also called the cochlear duct. That's where your organ of Corti, your spiral organ for hearing, is located. So this is a detail of your spiral organ and the ducts of the cochlea. Okay. Here's your hair cells, perilymph, and endolymph in the cochlear duct. So when you produce sound waves, okay, the best way to do it is do an illustration. You can see plucking a string makes it vibrate. That transfers energy that radiates through these air molecules. Okay, and when you see these air molecules, you can see in figure B, the frequency will equal the number of cycles per second. That's measured in hertzes. So, of course, that's going to be determined also by the wavelength. The wavelength is each one of these top waves is called, this is where it peaks, okay? That's called the peak, and then the bottom wave is called the trough, okay? So one peak and one trough, okay, is one wavelength. So from here to here is a wavelength. From here, okay. Right here, I may put it in dark here. So a higher frequency will have a basically the shorter wavelength will have a higher frequency, and you can see the pressure, the molecules of pressure. These are the ones where you see the high number of dots. That's a high density pressure. Those correspond to the peaks, and then the low density molecules where you don't see any dots. That's your troughs of your wavelength. So your pitch or frequency equals the number of cycles in 1 second measured in hertzes. So low frequencies and high frequencies. That's what we call pitches of sound. Amplitude or the loudness equals the difference between the high areas and the low pressure areas. That's measured in decibels. So you can see this in figure C. You can see a fairly low decibel reading in that first image in figure C. The second image will show a much higher amplitude or a higher loudness. If it's a difference between the high and low pressure areas. So those are the properties of sound: loudness and pitch. We have a loudness greater than, let's say, 90 decibels is what the USDA calls dangerous to your hearing. And eventually that can cause what we call a sensory neural deafness. So plucking a string makes it vibrate, transfers energy that radiates through the air molecules. You can see the pitch or frequency is equal to the number of cycles per second. So these would have the, in a low frequency sound wave, the high density molecules, the high pressure will be represented by the peaks, and the troughs will be the low density molecules. And then a higher frequency cycle per second, you'd see it look like this in figure B. Okay, even though it's, and then loudness, which is the amplitude of the sound wave, equals the difference between the high pressure and the low pressure. And that's measured in decibels after Alexander Graham Bell. Anything above 90 decibels is thought to be, is damaging. It destroys, or it produces damage to your hair cells in your organ of Corti of your cochlea. So if you look at this illustration, this kind of takes us through the process of hearing. Let's kind of go through this. First, when sound waves strike your tympanic membrane, that movement of that tympanic membrane will then move the auditory ossicle bones, your malleus, incus, and stapes, which are just sitting next to each other. They're not fused to each other, though. They vibrate against each other. What that produces, it basically increases the intensity. So that vibration eventually will reach the oval window. That produces pressure waves in the perilymph of the scala vestibule, as you can see from the black arrow. Okay. and eventually it curves around and goes into the scatotympany. Okay, so those vibrations of the oval window will produce pressure changes, pressure waves in the perilymph of the scatovestibule, as well as the endolymph of the cochlear duct. Okay, which then vibrates the basal membrane. The basal membrane is a boundary between the cochlear duct and the scatotympany. So pressure waves from high frequency sound waves will travel only a short distance, okay, into the cochlea. And they cause the basal membrane to vibrate where it is narrow and stiff. But as the cochlea extends toward the middle of the cochlea, the basal membrane becomes much more flexible, okay? So your pressure waves from your low frequency sound waves, they'll travel further into the cochlea, okay? And they cause the basal membrane to vibrate where the basal membrane is wider and more flexible. This is the shape of the basal membrane and how it varies, okay? So at the very beginning, the proximal end is very narrow and stiff, okay? At the highest, of course, is 20,000 hertz, and towards the end of the basal membrane, toward the tip of the cochlea, it's much wider and flexible. Low frequency sound waves are usually not lost in hearing loss, in sensory neural hearing loss, because they're at the very tip of the cochlea, whereas your high frequencies are lost usually when you have neural deafness. Those are the first to be lost. Helicotrema is where the scatovestibule and the scatotympany meet. But remember, they're composed of perilymph, and the cochlear duct contains endolymph. So your pressure waves from your low frequency sounds travel further into the cochlea, and they cause the basal membrane to vibrate where it's wide and flexible. There's the shape of the basal membrane as it varies. 20 hertz, those are the lowest frequencies, and the highest frequencies are up to 20,000 hertz. That's our range of hearing, between 20 to 20,000 hertz per second, vibrations per second. And here's a magnification showing you the spiral organ with their hair cells. Okay, and then the basal membrane at the bottom. Okay, the tectorial membrane is what overlaps the hair cells. And this is just another close-up. Okay, so the basal membrane will move up toward the tectorial membrane, bending the stereocilia toward the tallest stereocilium. Bending the stereocilia opens up the potassium channels that then depolarize a hair cell. Now you've got a depolarized hair cell that releases neurotransmitters that triggers an action potential in the axon of the cochlear nerve. Remember, the brain can only read action potentials. So first, the hair cell, when there's no sound, they're straight up. Okay. So all of a sudden, sound occurs. The basal membrane moves up toward the tectorial membrane, bending the stereocilia, okay, toward the tallest stereocilium. So bending these stereocilia opens up the potassium channels that depolarizes the hair cell. Now your depolarized hair cell will release neurotransmitters that triggers an action potential, which will travel through the axons of the cochlear nerve. So here's a micrograph showing you the stereocilia and the tip links. So hearing, first you have your action potentials propagating through axons of the cochlear portion of your vestibulocochlear nerve to the cochlear nuclei at the medulla pons junction. Okay. Then you got a second order neuron where you have the axons from the cochlear nuclei in the medulla, and the cochlear portion of the vestibulocochlear nerve, then they take it to the cochlear nuclei at the medulla pons junction. Okay, that's what you see in this first image here. Axons from the cochlear nuclei, then they synapse on the superior olivary nucleus in the pons. And then you have your third order neuron where you have the auditory stimuli, are then sent to the inferior colliculus in the midbrain. Remember, they form part of that corpora quadrigemini. So you've got 3 neurons now, and then your 4th order neuron then goes to the medial geniculate nucleus of the thalamus. Remember, you had visual information going to the lateral geniculate nucleus of the thalamus. Okay, so now you've got now 1, 2, 3. Your 4th order neuron goes to the medial geniculate nucleus of the thalamus. It's relayed. Then the 5th order nuclei. The thalamus then stimulates neurons of the primary auditory cortex and the temporal lobe, superior portion of the temporal lobe, and that's where the sensation is basically received. So this is just another map showing you how the sound is eventually reached. Okay, so first the ocean waves and your friend's laugh as he falls produces sound waves that enter the external auditory canal, as you see from the image. The sound waves then cause vibration of the tympanic membrane, auditory ossicles, and the oval window. Vibrations of the window then moves the perilymph and the endolymph and the cochlea, which in turn causes the basal membrane to vibrate. Then you get transduction occurring when the vibration of the basal membrane and endolymph depolarize the hair cells. That triggers an action potential in the cochlear neurons. Then you get the auditory signals, then travel through the axons of the vestibulocochlear nerve to the brainstem. Eventually, they reach the primary auditory cortex in the temporal lobe. Okay, and then that's when you get your conscious awareness and analysis of the sound will start. Okay, and the auditory cortex is the superior temporal lobe. But remember, they have these relays in these brainstem nuclei and also in the medulla. Okay, where you have the superior olivary nucleus and the inferior colliculus of the midbrain. This is again a location of the utricle and saccule and the relationship of the hair cells to the otolithic membrane and the macula. The macula is the expanded region, okay, of the vestibule where your hair cells are located. Remember, this is our static equilibrium. So here's your utricle and saccule in your vestibule. And here's your relationship of your hair cells to the otolithic membrane in a macula, which is, of course, in your vestibule. So when you produce action potentials, okay, when she's basically, now we're talking about the movement of the head. So the direction of the head tilt. When she's stationary, basically the hair cells are straight up, okay? And the tectatorial membrane is basically level, okay? Here's the conocilium, the hair cells, there's the vestibular nerve, okay? And these are the action potentials below, and the axons of the vestibular neurons. So when she tilts her head, okay, as the stereocilia bend toward the conocilium, the tallest one, that causes the hair cells to depolarize. Okay. And she's going to release more glutamate. Then she goes back to upright where it looks like the beginning. Okay, the action potentials are spaced apart. Then she turns her head the other way. Now the stereocilia are bent away from the cilium, the conocilium. The conocilium is the tallest stereocilia. Now the hair cells hyperpolarize, okay, and release less glutamate. Okay, so you'll see less of an action potential frequency. That's how we perceive balance, okay? And this just shows you another image showing you how transduction occurs for movements in different angles. When the endolymph, like when you move your head in a circle, okay? So the endolymph will lag behind and move in the opposite direction from the way the head turns. And that pushes on the cupula. This cupula is this looks like a cup that you see inside the endolymph, extending from the hair cells. Okay, so the head turns, that pushes the cupula, which depolarizes the hair cells and produces more glutamate. Okay, so the action potentials get closer together. Okay, when she stops turning her head, now the endolymph continues to move in the same direction as the head movement because of inertia. Okay, and it cause it hyperpolarization of the hair cells and produce less glutamate. So the action potentials are further and further apart. Here's the kinocilium, the stereocilia, and then the cupula. The whole thing is called the crista ampullaris. Remember, this is at the base of the semicircular canals, okay, of the ducts in the semicircular canals. The ducts are the canals with the fluid. The ducts are filled with the fluid, okay, the perilymph. So the cupula on the inside is surrounded by endolymph. The blue is the endolymph. So this is next figure shows transduction for angular rotation. So the head is stationary. Now the endolymph lags behind and moves in the opposite direction from the way the head turns. Okay, the pushes on the cupula, which then depolarizes the hair cells and produces more glutamate. So the action potentials get closer together. Okay. So she's moving her head to in this direction by the arrow in the figure. Okay. So the endolymph will lag behind and move in the opposite direction from the way the head turns because of the inertia. Now pushes on the cupula, as you can see in the image. Okay. In the second image. Okay. Which then depolarizes the hair cells and produces more glutamate, so the action potentials get closer together. Okay. And then when the head stops turning, the endolymph will continue to move because of inertia in the same direction as the head was turned. Okay, and that will cause hyperpolarization in the hair cells and produce less glutamate. So your action potentials will propagate to the vestibular nuclei located in the medulla pons junction, as you can see from your image. Okay. Then the cerebellum and the spinal cord will coordinate the muscle movement that maintains balance in response to this head movement. So once that message goes to the cerebellum, receive message from the cerebellum, you also have the cranial nerves, 3, 4, and 6. So the cranial nuclei will coordinate the eye movement in response to the head movement. Okay, that's the reason you have relays to those nuclei, 3, 4, and 6. But the cerebellum will also help to coordinate the muscle movement that maintains balance in response to this head movement. So eventually, once it reaches the thalamus, the vestibular nuclei then relay the signal simultaneously to several areas of the central nervous system. One is the thalamus, and then the inferior parietal lobe for conscious awareness of head position and movement. The cranial nuclei will then coordinate eye movement in response to the head movement, and the cerebellum and spinal cord to coordinate the muscle movement will also help maintain balance in response to this head movement. So that's basically your pathway for how the sense organs work together.